Hello everyone, my name is Xiangting Wang and I'm from the first affiliated hospital of the University of Science and Technology of China. Before I go to my talk, I want to express my great appreciation to the conference committee for their invitation and giving me this opportunity to share a recent work from my laboratory about the roles of long non-coding RNA alternative promoter variants in cancer. As we all know now, our genome contains a huge number of non-coding regions, and many of these regions are capable to be transcribed into non-coding RNAs, including the long non-coding RNA, a set of heterogeneous genes that are longer than 200 nucleotides. During the past 20 years investigation, it is clear that LNC-RNAs play critical roles in cancer, and they are involved in almost every aspect during the cancer initiation and development through diverse molecular mechanisms. Recently, more and more studies have found that LNC-RNAs contain abundant and diverse variants. For example, in a work that we published on genome research in the year of 2017, we found that about half amount of the total LNC-RNAs from the rhesus monkey brain have more than 2 transcriptional start sites. In another word, these long non-coding RNAs use alternative promoter strategy to generate different RNA variants. However, how these alternative promoter-derived LNCR variants function is unclear. To explore the function of LNCRA alternative promoter or AP variants, we took advantage of the cancer biology study because we have many excellent cancer cell lines to do the mechanism study. We paid attention to a pair of AP variants derived from link 00887, which is a LNC-RNA located on human chromosome 3 and has been previously shown as a hypoxia-induced RNA gene. We found that 887 contains different variants. In the tongue cancers, the two main variants of 887 are alternative promoter variants. We named the short one as 887S and the long one as 887L. Due to the alternative promoter usage, the promoter region of S retains two hypoxia response elements, or simply called HRE sites. Well, the L has no such elements in its promoter region. The difference of HRE size make 887S as a hypoxia-inducible gene, while 887L shows similar expression levels under both normoxia and hypoxia conditions in multiple TSCC cell lines. Additionally, we found that the two variants are both upregulated in TSCC cancer cells. Our next experiment was to test the biological function of 887S and L on tongue cancer cells. To our surprise, though both variants are upregulated in tongue cancer cells, knockdown of 887S remarkably promoted tumor progression in TSCC 15 cells observed by colony formation assay and transwell assay. While knockdown of 887L significantly inhibited tumor progression in TSCC 15 cells by the two assays. To confirm that these effects were dependent on the investigative variants, we also restored their expression in the corresponding knockout or knockdown cells and found that both overexpression could significantly reverse the effects caused by 887S knockout or 887L knockdown. Consistently with the colony formation and transfer acids modulating the expression level of 887S or 887L, dramatically affected TSCC growth in the opposite direction by xenograph experiments. Next, we want to identify the 887S and L-regulated downstream target genes. We performed the RNA-seq in the same knockout or knockdown cells I showed in the previous slide. Among the top regulated genes, we found carbonic anhydrase 9, or simply called C9, is a common downstream target of 887S and L, but in the opposite regulatory manner. C9 is a well-known hypoxia-induced gene. It plays critical roles in promoting tumor progression and hypoxia adaptation in many types of cancers. Our next question is, what are the regulatory mechanisms of LNCRA variants 887S and 887L on C9? Firstly, we conduct the C9 promoter luciferase reporter assay and show that both 887L and S regulate C9 at the transcriptional level. Since C9 has been widely reported to be induced by transcriptional factor HIF1 alpha under hypoxia treatment, we then performed RIP assay to test whether the two variants can bind to HIF1 alpha. Our results here show that only L but not S can interact with HIF1 alpha under hypoxia treatment. Next, we performed CHIRP PCR experiment to test whether 887L can bind to the C9 promoter. We also performed HIF assay to test whether 887L may affect the recruitment of HIF1 alpha to the C9 promoter. As expected, we show that 887S is indeed a critical factor to facilitate hypoxia-induced HIF1 alpha recruitment to the C9 promoter, and 887S as a LNCRA that does not interact with HIF1 alpha has no effect on HIF1 alpha recruitment to the C9 promoter. Now the question left to us is, what is the regulatory mechanism of 887S on C9 expression? We designed an open-ended RNA pull-down mass spectrum experiment. From the candidate 887 interacting proteins, we focused on DMT1 because of two reasons. One, DMT1 and demethylation are well-known negative regulatory mechanism for the transcriptional regulation. And two, there were two reports that had shown C9 may be regulated by demethylation. Using RNA pull-down following Western blotting and RIP assay, we confirmed the hypoxia-enhanced interaction between 887 and DMT1. To test whether 887S regulates C9 through DMT1-mediated demethylation, we first tested whether 887S and DMT1 can bind to the reported CG size of C9 by using 887S CHIRP and DMT1 PIP assays. We found that both 887S and DMT1 exhibited a hypoxia-enhanced interaction with the CG size on C9 promoter. Next, we want to test the requirement of 887S on DMT1 recruitment. we conducted another DMT1 triple by using the 87S knockout lines. Our results show that the recruitment of DMT1 to the CGCG sites of CA9 promoter was abolished in this 87S knockout cells. In addition, we wanted to test whether the 87-mediated C9 inhibition is through DMT1-mediated demethylation. To test this, we treated the 87S overexpressed TSCC cells with either siRNA against DMT1 or 5-AZA and found that 87S lost its inhibitory effect on C9 when DMT1 was knocked down or demethylation process was blocked by 5-AZA. Collectively, our results suggest that 87S mediates a DMT1 dependent inhibitory effect on C9 expression through demethylation in the presence of hypoxia. Until now, the observation that 88L-mediated HIF-1 alpha recruitment and 87S-mediated DMT1 recruitment were both hypoxia-induced. We then asked, is there a crosstalk between 87L and 87S-mediated pathways? Well, by performing the fractionation assay, we found that a good amount of both 87S and 87L translocate from nuclear plasma to the DNA bound fractions when the cells were treated by normoxia to switch to hypoxia condition. Well, when the cells received the hypoxia treatment, these two LNCR variants dissociate. These results suggest that the DNA binding ability of the two variants are regulated by their dynamic interaction. To test this hypothesis, we knocked down 87L and performed a series of experiments. Firstly, our data show that 87L knockdown dramatically increased the association of DMT1 and 87S, observed by both DMT1 drip assay and 87 RNA pull-down assay. Secondly, in order to investigate whether the above observed DMT1 recruitment was mediated through 87S, we further knocked down 87S in the two SH87L lines and performed DMT1 chip assay. Our data show that DMT1 recruitment was completely abolished after 87S knockdown. Moreover, we also observed a concomitant increase of demethylation in the tested regions flanking the CGCG sites of C9 promoter after 87L knockdown. To summarize, we have deciphered how LNCR variants control tongue cancer cell progression and explored the fine regulation of C9 expression upon hypoxia. One thing I would like to emphasize is that although the modulation of 887S shows a tumor suppressor effect during the cancer development, the integrated role of 887S together with 887L may help the cancer cells survive because the inhibitory role of 87S can keep the C9 at the relatively controllable level. Otherwise, the uncontrolled induction of C9 may induce cytotoxic effect to the cancer cell due to the shortage of nutrients. Well, This work was mainly done by my former student, Tao Shen, and I also want to thank our collaborators for this work, as well as all the members from my lab. Thanks for your attention.